It's my pleasure to present our work entitled The Role of Receptor Tyrosine Kinase, ROR2 in STEMNESS and Immune Invasion. Prostate cancer is characterized as an immunologically cold tumor with the loss of effector CD8 T cells and expansion of M2 macrophages and Treg. There's plenty of data that demonstrate negative association between the cancer stem cell phenotypes and cytotoxic T cell response and anti-cancer immunity in different cancers. As a consequence of powerful anti-androgen therapies, a subset of prostate cancer tumors become non-AR driven with the cancer stem cell phenotype. So we asked this question if this gain of cancer stem cell properties are associated with the immune suppression in this subset of patient or not. Previously, our lab generated ENS-resistant cell line with the low AR activity and enrichment of the cancer stem cell phenotype, capturing what is observed in the patient. So we use this cell line as a model, and gene set enrichment analysis suggests that not only STEMNESS and neural pathway are upregulated in these non-AR driven cell line compared to CRPC, but also enrichment of immune response. So as a baseline, we expose this cell line with the activated CD8 T cells and measure T cell mediated tumor cell killing, which is consistent with the hypothesis that non-AR driven cell lines are more resistant to immune cell killing compared to 16D CRPC. So as development of these phenotypes is not accompanied by many genomic changes, the field has been looking at the transcription factors and epigenetic regulators of these phenotypes. However, we wanted to see how kinase signaling feeds into the regulation of these proteins and survey receptor tyrosine kinases. And the advantage of receptor tyrosine kinase is that they are easily accessible and more druggable. Therefore, we integrate different RNA-seq data of metastatic castration-resistant prostate cancer patients and segregate them based on AR and NEPC score and categorize them in three groups: double negative, NEPC, and ARPC. And we noticed that among those patients who lost AR activity, whether double negative or NEPC, ROR2 is highly upregulated compared to other RTKs. So ROR2 is a receptor tyrosine kinase, and it's involved with non-canonical Wnt signaling. Active Wnt-ROR signaling has been linked to processes driving tumor development and progression, such as proliferation, survival, invasion, and therapy resistance. And similar to patient samples, prostate cancer cell lines which lost AR activity express higher level of ROR2 compared to AR-dependent cell lines. And using RNA-seq and gene set enrichment analysis suggests that the majority of pathways which are related to ROR2 expression are involved with STEMNESS and immune response. Therefore, we started with the functionality assay, and we identified that loss and gain of ROR2 affect spheroid formation ability along with ALDH activity, suggesting that ROR2 induced STEMNESS. Moreover, knockdown ROR2 induced T cell mediated tumor cell killing in non-AR driven, whereas overexpression of ROR2 in CRPC decreased this. So this result supports the notion that ROR2 enhance tumor immunosuppressive function and give an advantage for immune evasion. Moreover, correlation studies revealed that ROR2 positively correlated with the reprogramming transcription factors such as SOX2, OCT4, and NANOG, and also with the immunosuppressive checkpoints such as PD-L1, while negatively correlated with the AR and AR activity genes. Interestingly, in mCRPC patient, three genes, ROR2, SOX2, and PD-L1, co-express with the cancer stem cell score. Therefore, we knocked down ROR2 and we saw down regulation of reprogramming transcription factors as well as PD-L1. And recent publication showed that SOX2 regulate PD-L1 expression in other cancer, such as glioblastoma and cervical cancer. So we were able to produce the same data in prostate cancer, and we identified that both downregulation of ROR2 or SOX2 decreased PD-L1 expression. Moreover, we know that hormone therapy increase the expression of reprogramming transcription factors as well as PD-L1. And we identified that knockdown ROR2 is sufficient to prevent upregulation of these genes under the androgen-deprived condition. ROR2 also is positively correlated with PD-L1 in other cancer types such as bladder, lung, and colorectal. So in the summary, we identified that non-AR-driven tumors have a high expression of ROR2 as well as cancer stem cell properties. Modulation of ROR2 expression influence cancer stem cell properties and immune invasion, and knockdown ROR2 downregulates SOX2 and PD-L1. And now we are investigating the mechanism by which ROR2 regulates SOX2 and downstream pathways. Thank you.
Hello, everyone. My name is Ryan. I'm from the Flannigan lab supervised by Dr. Ryan Flannigan and the multi-scale design lab supervised by Dr. Hongshen. I hope you enjoyed my presentation on deep learning-based automated sperm identification for non-obstructive azoospermia patients. So approximately 15% of couples experience infertility. Of the infertile men, 15% suffer from azoospermia, the most severe form of male infertility, where there are no detectable sperm in the ejaculate. Non-obstructive azoospermia, also known as NOAA, is caused by a defect in sperm production. To help these couples conceive via in vitro fertilization, they extract sperm directly from a testis biopsy, a process known as microsurgical testicular sperm extraction, or microtesty. Only 50% of microtesty retrievals are successful, which are then injected into a single egg via intracytoplasmic sperm injection with the aim of fertilization to achieve a pregnancy. So this leads to the clinical problem. As the success rate of microtesty for NOAA patients is only 50%, this leaves half of the couples with no options for fertility. The primary reason for this failure is the inability to find extremely rare sperm among the millions of testicular cells. Shown in the figure, classic IVF can be done by placing an egg in an environment with lots of sperm, but that is not possible for NOAA patients. Currently, it is very possible for andrologists to miss a few sperm while they spend hours manually searching through tens of thousands of microscopy fields in order to identify and retrieve some sperm cells for use in ICSI. Therefore, the success rate of IVF ICSI for NOAA patients is currently limited by human cognition and patience. Our solution uses machine learning to automate a systematic process that not only saves the andrologist's time, but also provides a more robust and consistent method to help find rare sperm. So previous work aiming to automate sperm counting via microscopy images include approaches that use traditional image analysis as well as machine learning. In traditional image analysis, they use thresholding, edge detection, elliptical mass, and other techniques to segment sperm. However, these approaches are typically not very robust. Recently, deep learning was used to directly detect sperm from microscopy images. These studies identify sperm in semen where there are few contaminant cells, but are unable to deal with complex tissue like testis biopsies that include background cells of multiple shapes and sizes. So to find rare sperm among testis biopsies, we first develop an AI algorithm to detect sperm in sperm-only images as a preliminary test. We use this to find the performance upper bound, test sample preparation methods, and optimize imaging magnification. We tested images at 20 times, 10 times, and 4 times magnification to determine the magnification that balances performance and imaging time, as 4 times magnification is 25 times faster than 20 times magnification by area. The training dataset will be acquired by staining purified sperm from a donor to create fluorescently labeled ground truths, which are then later mixed with testis cells to train the model to differentiate sperm from testis cells. We captured bright field and fluorescent images that are then fed into the UNet, a widely used convolutional neural network architecture in medical imaging to learn and identify sperm. So sperm instances will be identified using the watershed algorithm and evaluated through precision recall metrics and receiver operating characteristic curves by comparing the detected sperm locations with the ground truth. So when evaluating the model for its use case, the model will automatically detect and stop automated imaging when it finds a sperm, alerting the andrologist and communicating its location, who will then verify the sperm and then extract it for intracytoplasmic sperm injection. Although fixed sperm samples are imaged for the training set, the algorithm is capable of detecting live sperm as well. So some results. We have generated sperm-only training sets and performed tests at different common magnifications to determine the upper bound performance. Evaluating with our custom metric that compares the predicted and ground truth sperm locations, our model achieves 91% precision and 96% recall in finding sperm in microscopy bright field sperm-only images, where precision is the proportion of detected sperm that are actually sperm, and recall is the proportion of all sperm that were identified correctly. The figure demonstrates the slight differences in image quality and corresponding performance. The performance falls slightly at 10 times magnification and sharply drops at 4 times magnification. We continue to study at 10 times magnification for its imaging speed as well as reasonable performance. So then we train the model to find sperm among testis cells, and we were able to find 74% of all sperm with 83% precision during testing. Here's an example of the steps used to count the sperm. So first, the input image, top left, an A, is put through the model to output a probability distribution for the pixels shown in image C, which is then thresholded for a binary image with yellow pixels being sperm in image D. Then the watershed algorithm shown in image E isolates sperm instances that are then visualized with enclosing circles and are numbered in image F. Image B is the ground truth used to verify the predictions afterwards and also used for training sets. One interesting observation is that the model understands and looks for the tail as a large indicator of whether or not a cell is a sperm, as you can see some semblance of a cell in the predictions. So next, I'll show you a quick demo of our system running and on our microscopy software. The sperm count is on the left and the green circles are the predicted sperm. The demo is intentionally slowed down so you can kind of see the process, but will be much quicker during live operation. As a quick explanation, the imaging area, the bright field area, is fed to the system, which is sliced into a grid of patches for predicting and stitched back together for the watershed algorithm before circling each sperm instance. In a clinical workflow, the automated imaging will stop once it finds a single rare sperm. And then back to the presentation. So for rare sperm validation, finally, we validate our system using rare sperm data sets. Unlike previous tests, there are far less sperm and the area near the wall edges were removed to better resemble clinical imaging quality and sperm concentration seen by andrologists. Hence the improvement in performance, finding 84% of rare sperm with 86% precision. As you can see in the fluorescent labels, the number of training label sperm in a single image is a few hundred, while rare sperm samples could have a few or none among the hundreds of testis cells and debris. Thank you for watching my presentation. I hope you found it interesting. I'm open for questions now.
Hello, everyone. Amelia and I are students in the Wyatt Lab at the Vancouver Prostate Center, and it's a great pleasure today to present our work on the profiling of nucleosome positioning patterns in the circulating tumor DNA of metastatic prostate cancer patients. So first, some brief background. Cell-free DNA is fragmented cellular DNA that's released into blood when cells undergo apoptosis. And circulating tumor DNA, or ctDNA, is the tumor-derived portion of cell-free DNA, and it captures information about the tumor tissue. So there are a variety of clinical opportunities for ctDNA, and this includes prognostic and predictive biomarkers, which are mostly based on detecting genomic DNA alterations in the tumor. And with these capabilities, ctDNA assays offer a less invasive alternative to metastatic biopsy. And this is especially true in metastatic castrate-resistant prostate cancer, where metastases are often in the bone. However, there's a really relatively unexplored characteristic that's unique to cell-free DNA, which could capture RNA-based information about the tumor and add clinical utility to ctDNA assays. And this is that the fragmentation of cell-free DNA is non-random. So during apoptosis, endonucleases in the cell tend to cleave DNA between the nucleosomes, and this results in short fragments with their endpoints mostly in the linker regions. And whole genome sequencing can capture these fragmentation patterns in cell-free DNA, and then this can be used to infer the positions of nucleosomes in cells of origin. Nucleosome positioning in gene transcription start sites, or TSS, are associated with gene expression. So it's thought that in highly expressed genes, the nucleosomes at the TSS are pushed aside by transcriptional machinery. And this causes a really regular pattern of nucleosome positioning near the TSS. This lack of nucleosomes at the TSS also exposes the DNA to digestion by endonucleases, and this results in this decreased sequencing read depth. And in contrast, the genes that have low expression don't show this pattern. So this suggests that fragmentation patterns in ctDNA could reveal information about gene expression in tumor cells. So gene expression biomarkers have been used for tumor subtyping and prognosis, but currently this information can only be obtained from a tissue biopsy. So the question becomes, can we infer tumor gene expression from cell-free DNA? We performed ultra-deep whole genome sequencing of 62 cell-free DNA samples that had high ctDNA levels from 33 patients with MCRPC, and 13 of these patients had bulk RNA sequencing data from time-matched metastatic tissue biopsies, which we use to validate our cell-free DNA derived gene expression metric. So we calculated a nucleosome occupation metric based on cfDNA fragmentomics, and this is adapted from Snyder and colleagues. And we also looked at the sequencing read depth. And plotting these two metrics over a genomic region that had very consistent nucleosome positioning, we see this really clean, regular signal with the peaks corresponding to nucleosome positions. So first, we looked at how well cell-free DNA-derived nucleosome patterns at the transcription start sites reflect tumor gene expression. So we binned genes using expression data from patient-matched metastatic tissue, and then for each of these gene bins, we summed the cell-free DNA nucleosome metric and read depth around the TSS. So the sum of this area marked in gray serves as a summary metric for the strength of nucleosome positioning here, and we correlated this value with the tissue gene expression deciles.
This results in a very strong correlation. On the left, we see two orthogonal metrics to assess nucleosome positioning centered at the TSS. Each line represents the data from one gene expression bin, with highly expressed ones in red and the least in blue. In both methods, the most highly expressed genes in tissue also have the strongest evidence of expression in cell-free DNA. And on the right, we can see this correlation holds true for all patients that we have matched metastatic tissue by RNA sequencing. We then performed the same analysis, but at the first exon-intron junction, where nucleosomes are thought to impact transcript splicing. And here, there's also a strong correlation between nucleosome patterns and tissue gene expression, but not as strongly as what we've seen at the TSS. Profiling nucleosomes can also reveal clinically important information in transcription factor binding. One patient in our cohort was suspected to have developed a more aggressive androgen receptor-independent disease over treatment. He was given abiraterone and enzalutamide with no PSA response. Mutational profiling of his serial cell-free DNA sample revealed the emergence of a new set of mutations while the old set disappeared, indicating a shift in tumor lineage. To confirm that he had progressed to AR independence, we used our method to analyze AR binding sites across the genome, and we show that as the patient progresses, evidence of nucleosome depletion at AR binding sites decreases and disappears. This supports the idea that AR no longer binds to those regions in AR-independent disease, so that nucleosomes are able to occupy those areas. This case shows that we can use ctDNA to monitor changes in tumor lineage and cancer-specific transcription factor activities. To increase clinical applicability, we're developing a targeted sequencing panel to capture the information as we did with whole genome sequencing. We also are performing the analysis on more subtypes of prostate and bladder cancers. But there are caveats to this method. So the signal quality is heterogeneous across the genome, so it's unclear whether querying one single gene would provide us very reliable information. So to summarize our work, ctDNA nucleosome profiling could proxy MNase and ATAC-seq to identify locations of nucleosomes in the genome. We show that ctDNA-inferred nucleosome organization at transcription regulation sites have strong correlation with metastatic tissue transcriptomes. These results are really exciting because fragmentomics are a unique advantage of cell-free DNA analysis that is not possible with genomic sequencing of tissue. It is possible that with one single well-designed targeted sequencing panel, we could get both genomic and transcriptomic information from ctDNA to aid in clinical decisions. With that, we want to thank all members at the WildLab and also other collaborators, as well as patients and families. Thank you all for having us today, and we're happy to take questions.
Yeah, good morning, everyone, and thank you for having me here. And I'm going to present my work on ASCL1 and how it facilitates neuronal lineage reprogramming to remodeling of chromatin landscape. So to begin with, the mainstream target of prostate cancer is androgen receptor, and we have seen more potent AR pathway inhibitors developed, like enzalutamide and abiraterone, to target the disease. But we have seen the emergence of a more aggressive neuronal-like phenotype termed treatment-induced NEPC. Other than the few genomic alterations between NEPC and CRPC, their genomic landscape is very similar, while they have extensive transcriptional profile, suggesting that most likely NEPC can result from an epigenetic plasticity. So using RNA-seq and ATAC-seq, we started interrogating the changes in chromatin landscape of CRPC in response to enzalutamide treatment. And we captured that enzalutamide can induce chromatin accessibility as early as 3 days after treatment with enzalutamide, but we saw this drastic expansion of the chromatin after 10 days of treatment. Mapping accessibility peaks and performing gene set enrichment analysis helped us to identify early activation of STEMNESS or plasticity pathway as early as 3 days post-treatment, following by activation of neuronal programming. This is while inhibition or suppression of canonical AR also started early on and continued to 10 days of treatment. So I wanted to see whether these new untangled chromatin as a result of enzalutamide treatment can support a neuronal phenotype. So we took this unique 10 days peaks and overlaid it with our 42D ENS-resistant and de novo NCI NEPC cell lines, and saw about 75% overlap with the 42D and about 25% with the NCI. To identify what might be the regulators, we performed motif enrichment analysis and identified ASCL1 motif to be highly enriched in both the regions that were shared between the 3 different samples and in the new regions that were gained accessibility in NEPC cell lines. In fact, early epigenomic alterations increased accessibility of ASCL1 motif as early as 3 days, and this enrichment in accessibility further continues into 10 days of treatment. ASCL1 expression and activity rapidly increases after treatment with enzalutamide and is very high in NEPC cell lines. So ASCL1 is a neuronal transcription factor. It's known for its role in neurogenesis, both during normal development of neurons, and it has been developed, its role has been developed very important to be both sufficient and necessary in other cancers like glioblastoma and small cell lung cancer. It's also very highly expressed and active in NEPC patients. So taking the unbiased approach, we knocked down ASCL1 in NEPC cell line and identified downregulation of pathways related to proliferation and neuronal differentiation following the knockdown of ASCL1. We further, to confirm this result, looked closer at specific neuronal genes, and here we show the regulation of the expression of these neuronal markers following targeting ASCL1. This loss of neuronal phenotype coincided with reduced proliferation in vitro and reduced tumor growth in vivo. So we mapped the genome-wide occupancy of ASCL1 in NEPC cell lines, and we identified that ASCL1 binding sites were enriched for neuronal genes. That was expected, but interestingly, we identified that ASCL1 syndrome is enriched for targets of PRC2 complex. Knockdown of ASCL1 do not affect the expression of any of the members of this complex. So we next look at H3K27 methylation, a surrogate to measure EZH2 activity, and observed that knockdown of ASCL1 result in global reduction of the cystone mark. In agreement with this, the 40% co-cubite site by ASCL1 and H3K27 methylation completely lost the cystone mark after knockdown of ASCL1. And these regions, as expected, were enriched for PRC2 targets, but also some luminal and basal pathways showed up. So we observed that pathways that were typically silenced by EZH2 initially were reactivating, and knockdown of ASCL1 was mimicking this reactivation of the adenocarcinoma, or the activation of pathways related to androgen canonical signaling. The switch back to the AR-dependent phenotype was further supported by a tag-stick where the 42D resistant cell line after knockdown of ASCL1 highly resembled the chromatin architecture of CRPC cell lines. These regions that were lost their accessibility following knockdown were majority enhancer regions involved in neuronal differentiation, while the regions that remained their accessibility and remained open were mapped equally to promoter enhancer enriched for canonical AR signaling. So to summarize, we discovered that ASCL1 is a potent regulator of NEPC, and that loss of ASCL1 in treatment-induced NEPC alters epigenetic reprogramming via EZH2 and switch the chromatin landscape to an adenocarcinoma. This reactivation of AR-driven state can provide new strategies for therapeutic intervention. Thank you so much for listening to my talk, and I'm happy to answer any questions.
So I will be presenting our ongoing work on how HOXB13 and switch-NIF complex alters the epigenetic landscape of prostate cancer. So I'm sure we all can agree that we have come a long way in treating prostate cancer, and especially with the approval of second-generation anti-androgen drugs, we have considerably improved the quality of the patient survival. But despite this, there is considerable number of patients who develop resistance to these drugs, and we definitely need new targets or new therapies to treat these CRPC patients. So our lab, we have been focusing on novel targets through which we can target pharmacologically. And one of the targets which we have been focusing on is HOXB13, which we heard in the keynote lecture speaker how extensively it is reprogramming the ER system. Before that, HOXB13 belongs to a HOX domain family of proteins, which has a highly conserved DNA binding domain. And it's very important in the embryonic development stages. And HOXB13 particularly is important for the development of urogenital sinuses. Interestingly, unlike other HOX proteins, HOXB13 expression continues throughout the adult life and remains primarily localized to the prostatic epithelial cells. As we can see here from the GTEC expression, while it is not expressed in any other tissues, prostate seems to have very high expression, and this expression continues even through prostate cancer. Recently, many studies have shown that it is important for prostate development and reprograms the ER system. The point which I want to focus here is all the studies which we have studied about HOXB13 is its role as a co-regulator for the ER gene expression or ER signaling pathway. Apart from that, hardly anything is known. So we focused on understanding HOXB13 more. So we looked at the depth map screening, and we saw that HOXB13 was essential only for prostatic epithelial cells, whereas prostate cancer cells, whereas it had no effect on other cancers. Bethel from our lab extensively validated these results in multiple prostate cancer cell lines. Much to our astonishment, it was essential only for prostate cancer cells, but had no effect on other cells. And interestingly, we saw that HOXB13 was also essential for PC3 cells, which we know which does not have air. So this not just the cell line, even the in vivo models show that knockdown of HOXB13 completely abolished any tumor growth. This convinced us that that HOXB13 definitely has a role other than regulating the ER transcriptional machinery. So, which brings us to our hypothesis that we hypothesize that HOXB13 interacts with specific co-regulatory factors in the tumor, which promotes prostate cancer growth. To understand these co-regulatory factors, what I did was we did RIME. So RIME is a proteomics approach which we use cross-linked prostate cancer cells and pulled down with OBSTRUCTIVE specific antibody and submitted to mass spectrometry. Here I'm showing results from PC3 and LN-cap cells, which shows a specific proteins which are seen in different cells, like while NKX and AR were abundant in LN-cap cells, they were not seen in prostate cancer cells, in PC3 cells. And among all these, what we found was SMARCD2, which is a component of switch-NIF complex, and this is what we have been focusing on. On further validation, we found SMARCD2 to interact with HOXB13 via co-IP as well as PLA. As you can see, we see good PLA interaction in LN-cap cells, but nothing in DU145 cells, which does not have endogenous HOXB13 expression. With this, we wanted to see what this interaction of HOXB13 and SMARCD2, does it have any clinical relevance? First, we looked at mRNA expression in TCGA cohort, and we saw a perfect correlation between expression of SMARCD2 and HOXB13. And similarly, one more targeted sequencing, CRISPR screen, which was done by Bengal in our lab, showed that SMARCD2 was essential to HOXB13 expressing cells, which kind of reiterates our point that SMARCD2 and HOXB13 may have clinical relevance. To further understand its role, we did HOXB13 ChIP-seq in both LN-cap and PC3 cells. While there was very little overlap between the HOXB13 binding sites, and as you can see here on with the canonical AR binding sites showed HOXB13 binding in LN-cap cells, and it was absent in PC3 cells, and there were new sites which were present in PC3 cells but not absent in LN-cap cells. But interestingly, the Giggle analysis showed that in both the cell line, SMARCD4 or the switch-NIF complex was highly enriched. The point to be noted is in LN-cap cells, the known ER co-regulators like ARID1A, NKX were enriched in LN-cap cells, but not in PC3 cells. With this, we think that HOXB13 may be functioning by recruiting a switch-NIF complex, which alters the epigenetic landscape, and this may be the mechanism through which HOXB13 is functioning. Just to summarize, we show that we know the HOXB13 shows a very specific tissue-specific expression and is selectively essential for both in vitro and in vivo growth of prostate cancer cells. And SMARCD2, a switch-NIF complex, is a co-regulator, a novel co-regulator of HOXB13, and probably alters the epigenetic landscape in prostate cancer. In the future work, we are in the process of investigating how HOXB13 and SMARCD2 interaction in clinical samples affect clinical prognosis, and also we are doing transcriptomic analysis after selective knockdown of HOXB13 and SMARCD2. Last but not least, I want to really thank everybody from LAC Lab and Hans and Stefan from mass spectrometry, pathology core, Sonia and Ladan have been really helpful for the clinical BLA. And last, I want to sneak in my son's picture, and thank you all for listening.